So thank you for joining into this session. So I would first like to thank the organizers for adapting to the current circumstances, and it's a pleasure to be virtually at this meeting. So the focus of my present today will be to present how Polyplus-transfection is supporting viral vector therapies from process development to commercialization. So who we are. So Polyplus-transfection is a French biotech company based in the European Bio Valley in Strasbourg. We are a leader. We are the first transfection provider for gene and cell therapy. We are experts. We have more than 20 years of expertise in transfection solutions. We're innovators. We focus on transfection solutions for therapy, bioproduction, and research. And last but not least, we are GMP pioneers. We are the first to have launched a GMP transfection reagent globally. What's our vision at Polyplus? So our vision is to provide life science companies around the world with goal-centered products and services to enable them to stay at the forefront in research and bioproduction, especially for the gene and cell therapy field. So, logically, our primary focus is to continuously innovate for the gene and cell therapy field. And as you know, viral vectors is the preferred delivery methods in the majority of ongoing nucleic acid therapy strategies to efficiently carry a gene of interest into target cells. By either direct injection into a patient, that's gene therapy, and or by first transducing patient isolated cells that then will be re-injected into a patient, and that's cell therapy. And for both strategies, these viral vectors are manufactured in human embryonic kidney cells using transient plasmid transfection. So to date, more than 400 recombinant viral vectors are in the clinical development phase, using both adeno-associated viruses and lentiviruses. And the use of viral vectors in the clinical setting has underlined the urgent need for process development systems that are capable of generating very large amounts of highly pure and controlled viral particles. And so manufacturing these large amounts of viral particles is challenging and costly. And this is why the two main hurdles that people are faced with in the viral vector manufacturing sector are increased viral vector productivity and meeting regulatory requirements. So how to increase viral vector productivity? It goes through optimization of the manufacturing process and switching to suspension systems, improving the transfection efficiency, and also improving the downstream steps. In terms of regulatory requirements, the focus is quality of these raw materials and securing the supply chain. So at Polyplus, our approach to support viral vector therapies from process development to commercialization is based on distinct pillars. We have 20 years of expertise in developing transfection reagents. We have a unique synthesis and formulation know-how that allows us to prepare to produce transfection reagents at different quality grades. We have a deep understanding of biology and application, which allows us to specifically develop transfection reagents optimized for different applications. And the focus, of course, today is gene therapy. In terms of supply and manufacturing, we also have an integrated platform that allows us to cover worldwide. And last but not least, we have an expert scientific and regulatory support team that is there to accompany each customer from process development to commercialization. So what's our core knowledge? So it's to produce viral vectors using transient transfection. So transient transfection means, in general, for all viral vectors, co-transfecting several plasmids in mammalian cells, mostly, so HEC293 cells and derivatives, to produce both lentivirus and AAVs. And what we do, our know-how is to develop transfection reagents that are specialized in improving delivery to cells and improving expression in these cells that will allow to improve viral yields. And increasing viral yields leads to improving the whole manufacturing process. So what are the current challenges of viral vector large-scale manufacturing, and how do we address them? So there are three main points I'd like to address today, and it's consistent and reproducible viral titers, focusing on AAV production and improving the scale-up, and last but not least, meeting the higher levels of regulatory compliance at earlier stages of clinical development. So first of all, in terms of consistent and reproducible viral titers. So at Polyplus, we provide a transfection reagent, PI-Pro, that is available at 3 different quality grades in order to fulfill the different quality requirements for process development, onto clinical trials, and up to commercialization. So the regular PI-Pro can be used to produce R&D grade viruses and for the manufacturing and process development. The intermediate PI-Pro HQ comes with additional quality controls that can be used to produce viruses for early clinical phases, and PI-Pro GMP that is used to produce viral vectors for late clinical phase and commercialization. And what we've nicely demonstrated is that with these different grades of transfection reagent, we're able to ensure that the same viral vector yields are obtained, and that consistent viral yields are obtained every time you synthesize a viral vector, both in adherent and suspension systems. So the fantastic thing about the PI-Pro transfection reagent portfolio is that you can select the quality grade that you need according to the quality requirements that you've established. Onwards to improving AAV production scale-up. Why? Why am I talking about AAV? Why am I focusing on AAV? Well, it's because what we clearly have identified in the gene and cell therapy field is that more and more gene therapies are based on the use of AAVs. And so we have AAV viral manufacturers that are coming to us with their cell systems, which often now switch to suspension systems. And what we're expecting actually is that in the near future, suspension systems will be widely adopted for large-scale industrial processes, as it is for protein and antibody manufacturing. And so with this in mind, our approach was to develop a transfection reagent specifically for large-scale transfection in suspension cells to produce AAVs. And that could be used at small scale during process development and up to large scale. And what we could achieve, our aim was to reach these objectives. So increase AAV titer yields, improve protocol for large-scale bioreactors, maintain the flexibility and reproducibility that manufacturers need to adapt to their system in place, and fulfill the quality requirements. And with that in mind, we recently launched Vectovir AAV, a transfection reagent that is specifically developed for large-scale transfection of suspension cells. And that comes with high performance, so superior yields for AAV production, scalability, it's developed for small to large-scale production, flexibility, it's compatible with commercial culture media, it's trustworthy because it comes from us, #1 transfection reagent supplier for gene and cell therapy. And so we know what we're doing. And it's GMP, in terms of GMP compliance, we're actively working on it, and the GMP grade should be available soon. And this is just to illustrate here on the nice graph, the data that you can obtain when you switch to Vectovir AAV. So I've talked to you about our PI Pro transfection reagent portfolio. And what you can nicely see is that Vectovir AAV is specifically developed for AAVs. And with that, we can even increase further the yields obtained. So we're really developing specific solutions to meet the current needs on the market. On towards high levels of regulatory compliance that are more and more required at early stages of clinical development. So the idea is to manufacture raw materials consistently and to meet the requirements in terms of tolerance, of purity, potency, and safety. And for this, we base ourselves on the existing guidelines. So what we know is that the quality of raw materials is determinant in the production of ATMPs. And it is clear that to minimize any variability due to the use of raw materials, it's important that these raw materials should be of the highest quality grade when it is available. And with that, you can mitigate your risks. So let's focus first on quality. So the idea is to decrease the risk associated with the use of raw materials. And what you want is reproducibility and lot-to-lot consistency. You want sterile raw materials that help you to decrease contamination risks in the cell culture. You want to minimize the variability of the raw material, and you want extensive traceability. And this is why by using a GMP-grade transfection reagent, you can fulfill these requirements. By it being compliant with GMP guidelines, you can ensure that it follows in terms of sterility, aseptic filling, that you have validated QCs according to the European Pharmacopoeia to assess the identity, activity, purity, and safety, and that you have a full documentation on the whole manufacturing process of the transfection reagent. So really, the ideal system to make sure that you meet regulatory requirements and that you decrease your risk to minimal is to use compliant raw materials and to secure them for your ATMP manufacturing. In terms of the second aspect is traceability. So on top of meeting quality requirements and using GMP compliant transfection reagents as we propose, the second aspect is traceability. And what we've recently launched is a GMP compliant residual test. Why? Because regulatory guidelines for the production of ATMPs indicate that residual levels of raw material used in the manufacturing process should be assessed, as well as the significance of the residual level detected. So ideally, that means that you should be able to determine how much of each critical raw material is still present, if any, in your final drug product. And this is where using a test as we have developed is ideal, because you can really mitigate your risk. You can detect and quantify reliably the transfection reagent throughout your manufacturing. And let's say, our custom-made test is adapted to your process so that we can ensure that we fit your requirements in terms of lowest limit of detection. And last but not least, securing supply. So in addition to securing quality, it's also required to secure the supply of raw materials. So this can be done by first qualifying the raw material suppliers. So raw material suppliers should be qualified as required by GMP guidelines. And so are we. The qualification and approval of these suppliers should be documented. You should be able to audit these key suppliers, and quality agreements should be in place. And an important tool to secure the supply process that I'd like to emphasize on is, of course, forecasting. And it's important to perform a precise forecast of the amount of raw material that you will need to reduce the risk related to the shortage of material. And this, of course, can be framed by signing a supply agreement. So we also think at Polyplus that it's very important to build as early on a strong and long-term relationship with key suppliers to have them on board when you move on to commercialization. And at Polyplus, we have a strong record of successful audits, and we are increasing our regulatory and quality assurance teams. And on top of that, we are also investing in this year to build new facilities in order to further support the worldwide growing demand of transfection reagents for virus manufacturing. And I'd like to finish with this slide, sort of a take-home message. Our winning strategy for gene therapy is to focus on the regulatory guidelines for viral vector manufacturing, so increased need for the quality and sustainable supply of raw materials for virus production, develop innovative transfection reagents to continuously help improve the viral vector, to help improve the production of viral vectors to meet the current needs. And this is why we launched Vector AAV, to really boost AAV large-scale manufacturing. And the next development on our side is, we're investing in facilities and human resources, and we're coming out with higher quality grades of Vector AAV. So the Vector AAV GMP is coming very soon. And I'd like to thank you for your attention, and I'm open to questions now. Thank you very much.